I'll use the envelope manufacturing as a case study. And I think it all really comes down, when you're talking about phase one, is to get a real balance. And I'll tell you what I mean by that. So if you look at phase one and phase three products, they're very different. And I think when considering what you want and what you need to make a phase one product versus a phase three, there are a lot of differences. One is, how much process development do you need? How much analytic development do you need? How much GMPs do you need? And Jim will talk about that more. How much you want to rely on the CMO? Is the CMO a good technical fit? Do they have the best facilities? And how much are you involved in the tech transfer and project management? How much quality you have? And when does it make sense to partner with industry? So we've had 2 successes with HIV envelope protein manufacturing post-RV144. That's not a lot of success, but we've had it, and we're trying to get as much as we can from these models. So Novartis / GSK, NIAID, the Gates Foundation, all working to make the 2 P5 proteins, and Dan Baruch, Crucell, J&J, NIAID supported to make the Clade C GP140 and a subsequent Mosaic GP140 Dan will talk about. But that still took 5 years. So most people now want to get phase 1 products into testing, or products into phase 1 testing. And we realize that we have to then look at the learning efficiencies and move forward and translate that in a way so that we could not have to wait 5 years for proteins again. And so, how do we do this? I hope I've given you enough to understand that you can't just flip it over the fence to the CMO, and GMPs are not the reason that we're getting hung up, the requirement for tox studies and all that. You just need to have something that's safe and characterized, and you need to find the balance for your product. ONV is a particularly challenging one, so the balance is a little bit harder to do. Some other phase 1 products may be easier to get into a vial. But, you know, and at the end of the day, you have to understand the process, and then you have to be able to work with the CMO, and you have to understand the characterization, and then you could tinker with that process. And as long as you are assuring that the product is sterile, and you could make something with consistency, I think you'll be in good shape to move some of the phase 1, some of your research candidates into phase 1 products.
We work in a very regulated environment. FDA's primary objective in reviewing an IND are in all phases of the investigation to assure safety and the rights of subjects, and in phase 2 and 3 to help assure that the quality of the scientific evaluation is adequate. We should design our products with safety in mind. And if possible, we should avoid some things that are going to be problematic. Novel adjuvants with no clinical safety data is going to delay your program, because you'll have to demonstrate a lot of safety data in animals before you can start to put a novel adjuvant, a truly novel adjuvant, into people. Other things to avoid: neurotropic or cardiotropic viruses, cell lines which have not been used for vaccines, or novel excipients or raw materials. One of the things I've learned over the years is that it's important, it can be important to have consultation and meetings with regulatory agencies to engage them in the development process. I like to use it to validate your development strategy, which an FDA and the European agencies are usually quite good at this. You go along with a plan. Don't go along and ask them what to do. Go along with what you want to do, what you think is defendable, and you can. And they're usually happy to give advice and consider your audience and their expectations. Like someone said to me the other day, you need to be the smartest person in the room when you go and talk to FDA. They're there to do a job. They're reasonable scientists. They're not experts in your product, but they're there to prevent disasters. And they're going to be there to minimise the risk to the population. So they have a job to do. Sometimes people come to me and say, FDA doesn't understand what we're doing. Well, I say then, you haven't explained it properly to them. So you need to explain what you want to do and why you want to do it.
I'm going to give you some key test cases that we've looked at and are not looking at, and so that you can see how things can be done, very much with an eye to early phase 1 experimental medicine studies. And when we talk about experimental medicine studies, we're not talking about not doing phase 1s. It's really more a shorthand way of saying doing studies that are to inform vaccine development rather than being on a conventional product pipeline that has a view of going from phase 1 to phase 2 or phase 3. Because much of what we look at, the majority of what we look at, we are expecting only to go to phase 1 and inform what we need to do to make a real-world HIV vaccine. So what do we mean by experimental medicine trials? So really, hypothesis testing in order to better develop vaccine in an iterative fashion and reduce the chances of late-stage failure in large clinical trials, but also to provide an environment where you can do very detailed, in-depth investigation. So I'm going to start with the first test case, and that's looking at DNA vaccines. So why are we interested in DNA vaccine approaches? The advantage here is essentially cheap and fast to manufacture, so at least 1/10 of the cost of taking a recombinant protein into a clinical trial. The disadvantage is obviously with a DNA-only approach is that they're considered to be relatively poor immunogens. So you would never go forward with a DNA strategy if your target product profile was to develop a real-world vaccine. But if your question is, can we come up with something that would allow us to induce immune responses that, for example, would allow us to screen multiple immunogens, look at B-cell responses, there is a potential plausibility that this approach would be a way to go forward. So this is the test case that currently we're looking at, doing a trial called DNA-VAC. And the benchmark that we've set ourselves in order to be able to screen immunogens for immunogenicity is to have a high level of seroconversion, to have enrichment of antigen-specific plasmablasts and memory B cells for repertoire analysis and next-generation sequencing. So this is an ideal framework for an experimental medicine approach. The second test case that we're starting to explore is using envelope proteins. And ultimately, we probably need to be looking in the tens or more of envelope proteins to be able to explore all the competing hypotheses that are currently out there that for the approaches or the roadmaps that may need to be discovered to learn what it needs to make broadly neutralizing antibodies. So what are the potential ways that we could accelerate that? Well, now we're looking at making new envelope proteins. We're going for a bacterial artificial chromosome technology. This allows us to give us a rapid establishment of stable cell lines. We reckon we can save 6 months in terms of the generation of stable cell lines. We have adopted a generic manufacturing process. We're doing smaller batch production. And so all of these different steps that we gained from the first time doing this, we think we can cut the time down from discovery to the clinic from about 36 months to 24 months. So in terms of the lessons that we've learned, looking at this early phase of experimental medicine testing in phase 1 trials, we think that small experienced teams can be cost-effective. It does require good management, but it gives us rapid decision-making processes, shorter timelines, fewer tiers of administration, which means that often we can move quite quickly, and it gives us the flexibility to change our scientific priorities. And that is often important given the pace of change in terms of scientific progress. So we think that this model is appropriate for iterative experimental medicine trials.
Our vaccine program has been an NIH-funded program under a program called the Integrated Preclinical Clinical AIDS Vaccine Development Program since 2004. And in my view, I think NIH was visionary in developing these programs, essentially acknowledging that industry participation is critical in a vaccine development field. Otherwise, we could just purely be a vaccine research field and essentially have no product as a goal. But if we want to be a vaccine research and development field, we need industry participation, and preferably big pharma. And so the NIH established these programs as a way of incentivizing industry, to give grants to academic investigators to seek out industry partners to develop programs together with them. So the vaccine that we're moving forward with is a multivalent vaccine. Again, each component of this vaccine is informed by a well-powered NHP challenge study. Priming with Ad26 vectors expressing HIV mosaic antigens, and boosting with either Ad or MVA vectors with a purified GP140 protein. The manufacturing of this vaccine was exceedingly complicated, and within this manufacturing program is probably where most people would consider that our program shifted, at least partially, from research-oriented development to a product-oriented development. And the success of this program, of the manufacturing program, was entirely dependent on the ability to solve problems along the way, essentially problems that could not have been predicted in advance. Critical for moving this program forward was the organization of this program as a pharmaceutical development program with very substantial input of Janssen staff. There's a compound development team. Each of the products has a steering group with many different inputs, and our group and DAIDS were embedded into this very large matrix of teams. And I should mention that the entire commitment from Janssen has required over 50 FTEs to make these products happen. We're currently in phase 1 and 2A clinical trials, and a phase 2A regimen down selection study has been fully enrolled in 400 subjects in the US, East Africa, South Africa, and Southeast Asia to help with regimen selection. In addition, there will be a series of studies and ancillary studies in 2016 to help refine some technical components of the vaccine, with the hope that the vaccine, if it reaches predetermined go-no-go decisions, will move into efficacy trials, both in Africa as well as in US, Latin America, Asia, and Europe. One of many critical features that allowed a transition from research-oriented to product-oriented development was a committed industry partner. And I think that can't be overstated in terms of the importance of that. Also critical for our program was very strong personal relationships that have been built over many years of collaboration. With any academic industry partnership, there's a lot of give and take. And we agree on many things, and we've disagreed on some things, but we've always been able to talk through issues and work things out. And I think this strong personal relationships between government, academia, and industry is a critical foundation for these type of programs. Also, there has to be a vision. I think it's critical for an industry partner to feel that there's a downstream potential for the product, which means that manufacturing at scale has to be feasible, or at least aspirational. There has to be a business plan that's viable, and the legal issues have to be solvable. These are issues that we as academics don't think about, or don't often think about, but a partnership with an industry partner would not be possible without these elements. And then also, I think critical for a product-oriented program is that the industry partner is a regulatory sponsor and is the leader and driver of the clinical development, but yet the academic groups still have much to offer, and so the collaborations with academia and government continue, and we hope they will continue to continue.
As an academic who's interested in product development, it's Tim Cardozo back here, I'm sold on the idea that was mentioned several times that an industry partner is extremely important. Industry in general and the kind of industry size that would be necessary for this are not going to be interested in a product until it's almost a product. But almost everything in the academic world is nowhere near there. So how do you bridge that gap to get your committed industry partner?
In our experience, it has been a process over 10 years that has been essentially a mutual growth process that we think has been founded on data along the way. So small animal data leading to large animal data, leading to phase 1 clinical data. And then until very recently, then the support from NIH for these translational preclinical to clinical programs was critical, because certainly in those early years, it would have been very unlikely that an industry partner would have been willing to invest.
But in terms of this whole area of how and when to engage industry at early stages where there's a perceived higher level of risk is a really big issue. We've tried to approach it from a number of different angles. We've tried to engage in sort of umbrella agreements with companies that allow us to have discussions with them around things that are in development in the pre-competitive space, where they may not have the resources or the willingness to invest in it internally. But if we're able to position, we're able to put in some funding or establish collaborations for them that allow them to continue to work in a space and de-risk it in the near term, we can figure out a way to make it work for them that's at lower risk and lower cost to them. And so we've done this now with, I think, 3 or 4 companies.
Each company has different mechanisms, but it's sort of recognized that a lot of the, if not all or most of the innovation occurs outside the big pharma because they have limited ability to do novel and basic research. And so all the companies have set up in one form or another a way to engage with academics and small biotechs. In the case of Johnson, we have 4 innovation centers, one in Boston, one in San Francisco, one in San Diego, and one in London. And their goal is to search out novel, innovative approaches to things we're interested in and engage them early, either through a variety of mechanisms, investments, and if they happen to have a company, or early support of work or so on, not trying to jump in and manage the company or the academic, but rather to give them the freedom to work, but also we're involved early. So we do have mechanisms to engage, and I think one of the problems is that people don't know about these things.
In our experience, having really robust project management to pull together all of the pieces is absolutely critically important. And even if you've got it right in a regulatory perspective or in the manufacturing, unless you have that glue to pull it all together, it won't go efficiently or smoothly.
And having somebody whose sole job is to spend every day worrying about how they can move this further forward, how they can move it further forward faster, what is going to go wrong. The ideal person for this is a terrific organizer and a constant worrier. And that person ideally takes a lot of the weight off the back of the leading scientist.
Dan said something near the end of his talk that I thought, he said it very quietly, and I think maybe we missed it. He said, there's got to be a business plan for an HIV vaccine. And I think, drawing back to what Jerry said earlier, the business plan has to consider or propose that it's possible to make an HIV vaccine with a positive NPV. So that's one of the things that we've always struggled with when we talk to people, is when we're asked, okay, you have an HIV vaccine, what's the business plan to make this viable or to give you a positive NPV?
Suit. I think the science of the field has advanced to the point where the HIV vaccine field is worthy of true pharmaceutical investment. And we've seen that with one company, and hopefully we'll see that with others in the next few years. Time will tell. But I think that there are skills with how to interact productively with industry partners that no one ever teaches us in medical school or graduate school. And I also mentioned very quietly that there's always a lot of compromise and give and take. So a lot of, and I think that is critical for building the relationships to move something forward. And I can't stress how important the personal interactions are for academic industry relationships, at least in our experience. I think what academic groups can do is to present a good scientific case